Good afternoon, everyone. My name is Kumaran Ramamurthy, and it is my pleasure to introduce, on behalf of the Lambda Lunch Bacteriology Interest Group, Professor Peter Greenberg from the University of Washington School of Medicine. Professor Greenberg received his PhD from the University of Massachusetts and did his postdoctoral work at Harvard University with the legendary Woody Hastings. He was initially on the faculty of Cornell University and the University of Iowa, before moving to the University of Washington, where he has been for the last 10 years. Professor Greenberg has been a pioneer in the field of bacterial quorum-sensing, the mechanism by which gene expression is regulated in response to population density, typically by sensing the accumulation of a secreted chemical signal. In fact, he actually coined the term quorum-sensing in 1994 with Clay Fuqua and Steve Winans. At first, the idea that bacteria could communicate with each other using a chemical language was not widely accepted. Indeed, Professor Greenberg has put it more bluntly by saying most people thought it was nonsense that bacteria could communicate. However, today, understanding this phenomenon is at the heart of elucidating strategies to combat infections such as those that affect cystic fibrosis patients. As such, the Greenberg lab has been largely focusing on studying how the opportunistic pathogen, Pseudomonas aeruginosa, monitors the density of its population during infection and how it uses this information to form biofilms. The lab has reported, for example, how these molecules are synthesized, how they bind their cognate receptors, and how these binding events trigger gene expression. For his work, Professor Greenberg has received many awards. Most recently, he has been awarded, along with Bonnie Basler, the 2015 Shaw Prize from Hong Kong, which is affectionately called the Nobel Prize of the East. He was also elected to the National Academy of Sciences in 2004 and to the American Academy of Arts and Sciences in 2002. Please join me in welcoming Professor Greenberg to the NIH.
Okay, thanks for the great introduction, Kumar. It's an honor and a pleasure to be here. I was trying to remember the last time I was at NIH, and I remember I was here. That's about it. So today I want to tell you some things about sociality and bacteria, and I'm going to focus on what you just heard about, quorum sensing. And the story today, when I get to it, will be about how we're now able to do what I call social engineering experiments. I view myself like the government of Singapore, lording over bacteria, getting them to do what I want. And I've organized the talk. I just put this slide and a few other ones in. There'll be a short prologue where I'll introduce you to some of the luminaries in the area, the historical figures and how I got into it, and then an introduction and a review of a lot of work that we've done over the years, finally getting to some data, and then there'll be some conclusions and a short epilogue. So that's how we're organized. First, the prologue. I wanted to start with a picture of this guy, Albert St. Georgi, the Nobel laureate, and because I think this quote is prescient. Research is seeing what everyone else has seen and thinking what no one else has thought. And I don't entirely agree with this, but it's partially true, and it helps set the stage for how we develop this field. So I first was introduced to him as a student at Woods Hole in 1973. I was also introduced to this fellow, Ed Wilson. He's the father of a field he called sociobiology. And what he means by sociobiology is a study of the genetic basis and environmental influences on social activities. And he felt that sociology, social activities, should be the domain of the biologist. This was very controversial when he introduced this idea in the 70s, but it's become more and more accepted. And he views sociality simply as the sum of conflict and cooperation between two or more individuals. Wilson studied ants, and by watching them, he could see that sometimes ants in colonies were cooperating and sometimes they were in conflict. Well, I study bacteria, which are organisms like ants. They're also single cells. And the point is then that I hope I can convince you that cells in any context can be social. So over the years since I began and people didn't believe that quorum sensing existed, we've established a new field that we call sociomicrobiology, and now people accept this idea that bacteria are social entities and that whatever the selective pressures that have led to sociality in higher organisms, whatever those are, they're similar for bacteria. And we work on sociomicrobiology for several reasons. First, bacteria, I maybe have to convince a few people in this room that bacteria are wonderful models to understand the biology of almost anything, including sociality. We can do a grand social experiment with millions of individuals interacting on a plate on our benchtop. We can make mutations at will and see how those affect sociality. And we can even do evolution experiments over a period of a few weeks in the lab. So I've always been fascinated by bacteria. If you really want to understand bacteria, I hope to convince you you can't ignore their sociality. And there's this idea, as Kumar said, to develop new medicines to treat devastating bacterial infections that are difficult to control. Ideas that by targeting sociality you can do this. We'll see. Okay, on to the introduction. I'll introduce you to a couple of more luminaries. This is the, what did you call him, the famous Woody, the what?
Legendary.
The legendary Woody Hastings, my postdoctoral advisor. In the early 70s, he discovered, he loved everything luminescent. And he discovered that luminescent bacteria only made light as they grew in cultures and reached a high density. And he introduced a hypothesis he called the autoinducer hypothesis, that these bacteria were making some signal that went out into the environment, and when it reached a high enough concentration, it was recognized and it signaled the bacteria to make light. So they only made light at high density. And I became fascinated about this when I learned about it at Woods Hole. So there was St. Georgie, who was at Woods Hole. There was Wilson, who spends his summer at Woods Hole, and Hastings, who was there in the summer. And I decided this is what I wanted to work on. I never met this guy. I want to introduce him to you, though. Garrett Hardin. He introduced the idea of what's called, among other things, a tragedy of the commons and Darwin's dilemma. And I'll start with this little bit here. Faculty member at the University of Maryland, Professor Selterman, I read about this on like Yahoo News or something. Somebody wrote, "What kind of professor does this?" It was one of his students. And this is what Selterman did. He said, select whether you want 2 points or 6 points added onto your final paper grade. Easy. But there's a small catch. If more than 10% of the class selects 6 points, then no one gets any points. So there's some self-interest in selecting 6 points. But the whole group loses if too many people do this. This is a tragedy, and Selterman has been doing this for some 15 years, and only 1 year did the students cooperate enough to all get 2 points. Every other year they got nothing. There was an incentive to be greedy. So this leads me to Darwin's dilemma. I guess I should have a picture of Charles Darwin here, but I don't. Cheats that take advantage of cooperators should have a fitness advantage. They don't have to spend whatever resources are necessary to cooperate, but they can benefit from the group of cooperators. Tax cheats are my example. You know, I pay my taxes in Seattle and we get relatively decent roads and relatively decent policing. Cheats also get those things and they don't pay their taxes. So cooperation should be unstable. It should be selected against if cheats have a fitness advantage. This is Darwin's dilemma. How does cooperation evolve? How is it stable? And so some definitions before I move on to my review of quorum sensing. These terms, I want to point out, all come from the field of population biology. I'm not making them up. Someone else made them up, and I have to use them. And that's my disclaimer, because in a group this big, at least one person will be indignant that I'm using terms that they view as anthropomorphic. They're not in the sense that they have specific definitions in biology. Cooperation is a behavior that provides a benefit to another individual, a recipient. A cheater, these are sometimes called freeloaders, is an individual who doesn't cooperate, who cooperates less than its fair share, but who can gain the benefit of others cooperating. So as I just described, cheaters have a fitness advantage over cooperators. I'll use this term restraint. It's a self-imposed cost to cheating. So sort of, you know, I have a moral urge to pay my taxes. Policing is also called punishment or sanctioning, and this involves the cooperators hindering the success of social cheats, and they do this at some expense to themselves. And I already introduced the tragedy of the commons. It's called A Few Other Things. It was introduced by Hardin. And it's depletion of a common resource by acting rationally in one's self-interest. So selecting 6 points over 2 points. Now the review. You can see I'm moving along, just in case you're already ready to leave. So I want to tell you a little bit about what I call acyl-HSL signaling. It's a kind of quorum sensing. It was the first to be described at a mechanistic level. And I'll concentrate on this opportunistic pathogen, Pseudomonas aeruginosa. And I'm into this review now of quorum sensing. Then I want to tell you about social cheating and P. aeruginosa and a tragedy of the commons, and then self-restraint and policing of social cheats. How is cooperation stabilized? And the social cheating part was done by these 3 individuals. Ajay Dandekar, who was a pulmonary fellow in my lab, he's now on the faculty in pulmonary medicine at Washington. Sudha Chugani, who was a longtime research associate in my lab. And I think this project finally drove her away, and she's now working in the informatics field. And Mei Wang, who was a visiting faculty member from a university in China. So quorum sensing is a type of bacterial cell-to-cell signaling, and it's used to control specific genes. It allows coordination of group activities. Group activities are underlined. That implies there must be some sort of social interactions. There are lots of different quorum sensing systems that we know about now. The most well-conserved, the most well-studied is the acyl-HSL type that occurs in proteobacteria, gram-negative bacteria. We now know of over 200 different species that use quorum sensing signals. So not all proteobacterial species do this, but many do. The signals are dedicated. They're made by the so-called LasI family of AHL synthases. There are co-evolved LasR transcription factors, signal receptors. And I mention this coevolution because in population biology, the coevolution of a signal and the receptor is a key to calling something a true signaling system or a true communication system. So this appears to be some sort of true signaling system. And these LasR-LasI systems regulate different things in different bacterial species. I'll have more to say about that in a minute. Okay, this is a Petri plate of colonies of Vibrio fischeri, woody Hastings luminescent bacterium. And the colonies on the plate expose the film. They make this blue light. And they only make blue light when they're at high population density. They're at high population density in colonies on plates. So somehow they know when their populations are high. And in the 80s, our lab and one other lab, Mike Silverman's lab in California, worked the details of how this operates out. And this cartoon, which doesn't show up too well here, was actually one made by my colleague Monty Bassler to sort of summarize what we've learned about quorum-sensing and Vibrio fischeri. So in very light blue here is the outline of a cell, and these are genes in the cell, genes for luminescence. So the LasI gene is the first gene in an operon, a group of 7 genes. Well, let's see, 1, 2, 3, 4, 5, 6. There's also a G that's not here that are involved in light production. And LasI generates an enzyme that makes a signal that can diffuse in and out of cells by passive diffusion. This is the AHL. I'll show you one of these in a minute. So when there aren't very many cells in a local environment, as they make it, it will flow out down its concentration gradient. It can accumulate, and it won't do anything. When there are cells packed close together, the concentration of the triangles can rise. And when it reaches about 5 nanomolar, it can interact with the LasR protein. This is a transcription factor, and when it's bound to the signal, it activates this cluster of genes, and the cells make light. So we know how, as Kumar told you, we figured out how these things make signals. We figured out how these things bind to the signals, how the promoter is activated, and all sorts of stuff that I'm not going to tell you about today, but led up to some really simple experiments. So, you know, you do work for a decade or two just to do with all sorts of sophisticated stuff, just to get to the point where you can do some simple experiments. So we'll see about those. This is the Vibrio fischeri AHL. This compound was uncovered by a colleague when I was at Cornell. He was at Ithaca College, Anatol Eberhard. It's an acylated amino acid. This is one of the 20 essentials. It's homoserine lactone. And the compound is made using a fatty acyl ACP as the fatty acid donor and S-adenosylmethionine as the amino acid donor. The amino acid part makes it relatively soluble in the aqueous phase. The fatty acid part lets it slip in and out of the membrane. So in the 1990s, we started to recognize that this signal and these systems weren't peculiar to Vibrio fischeri, that lots of bacteria made these signals. This just shows a smattering of some of the signals that have been discovered. One of the sports in my lab over the years has been discovering new versions of these signals. And so there are about 30 or 35 different ones known now. And the signals and their receptors are pretty specific for each other with some quirks to that. So there's a coevolution of signal and receptor. So what does quorum sensing control? So this brought us into what I call my transcriptomics era. This is now over with, and I'm really happy about that. Well, I already told you about light production in Vibrio fischeri. No individual makes enough light for any sort of biological detection. So nothing can see the light made by a single cell, but we can see the collective activity of the group. So I view this as a cooperative activity. And transcriptomics told us a very limited number of genes were regulated by quorum sensing in Vibrio fischeri, depending on the strain, 10 to 25 genes. In other bacteria, like the one I'm going to tell you about today, Pseudomonas, exoenzymes are controlled by quorum sensing. And again, the idea, which has been validated by experimentation, is that I'll use proteases as an example. So Pseudomonas can grow on milk protein, but it has to make a protease that's secreted into the environment. A single cell can't make enough protease to break proteins down into small enough pieces to get any value, to get the concentration of those amino acids up high enough to transport in and use as energy. But a pack of cells can make enough to gain benefit. So exoenzymes are what we call public goods. They're made by all the individuals and shared among them. Toxic compounds, like antibiotics, are often controlled by quorum sensing. Again, the idea is a single cell can't make enough antibiotic to kill anything, but a group of bacteria can make enough to protect itself from competitors. Antibiotics are excreted, so again, these are public goods or common goods. So now what about Pseudomonas? This is an opportunistic pathogen that senses cell density through quorum sensing. We discovered this in the early 90s. It infects all sorts of things, including people. And I've been particularly interested in the lung infections that people with cystic fibrosis get. So they live with these infections for decades. And quorum sensing was shown early on by others to be required at least to initiate infections. And the idea is that when Pseudomonas first enters a host, it will be at relatively low population density, so the quorum sensing signal concentration will be low. And in that state, there'll be little activation of whatever's controlled by quorum sensing. And we know that a battery of virulence genes are controlled by quorum sensing, and normal biofilm development is influenced by quorum sensing. So the bacteria can grow under the radar of the host, and when they accumulate to a high enough density in a local area, the signals, the stars here, will alert them that there are now enough to activate expression of what we have learned is a regulon of about 300 genes. And at that point, they can overwhelm host defense mechanisms. They've deprived the host of a chance to begin to react to low levels of these things. And the transcriptomics showed us that genes coding for secreted products or the production of secreted products are, I say, grossly overrepresented in the quorum sensing regulon. So most of the genes, at least the ones where we know the function, that are activated by quorum sensing are involved in putting something outside of the cell, things that could be public goods. Pseudomonas uses two quorum sensing signaling systems. I'll call them the C12 HSL system and the C4 system. And this shows, this is the C4. It's made by a LasI homolog we call LasI, and the transcription factor that responds to it is a LasR homolog called LasR. This is the C12 signal. It is made by LasI. LasR responds to this signal It doesn't respond to the C4 signal. And this will become important a little later. The LasR and I system regulates a whole bunch of things, including the RelR and I system. So we say that LasR and I is on top of the quorum-sensing signaling cascade. And for this reason, most people that have tried to develop small molecule inhibitors of quorum sensing have focused on blocking LasR. This may or may not have been an idea with wisdom, because it turns out that although LasR is a therapeutic target, LasR mutants are common in certain infections. And one infection that they accrue in over time is the infections of cystic fibrosis lungs. So people who've been colonized for some years with Pseudomonas, when we isolate the bacteria from their lungs, the bacteria have evolved and adapted to the lungs. And one thing that happens is that they accrue mutations in LasR. Well, why do they do this? I don't have the answer to that yet. I'll have more to say about it. They might be social cheats. They might be cheaters in among cooperators. And if that's true, then it proves that LasR is critical for these infections. Cheats only will occur when cooperation is essential. So it could be a free rider problem. And the idea is that quorum sensing defective individuals don't share in the production cost of public goods like secreted proteases and antibiotics, but again, they accrue the full benefit afforded to an individual member of the group. So I'm sort of pounding this in. Cheaters should cause a tragedy of the commons, but this clearly doesn't happen in chronic infections. So from a whole lot of experiments, we've learned that exoenzyme synthesis is primarily controlled by LasR, and exoenzymes are overrepresented in the LasR regulon. RelR, which is activated by LasR, controls the synthesis of toxic molecules. So Pseudomonas makes cyanide. Pathogenic bacteriologists see this as a virulence factor. And clearly it is, but it will affect all sorts of biological creatures. Diane Newman was here last week, right? And I don't know if she taught, she studies piocyan and among other things. And she views this as an electron shuttle. Pathogenic bacteriologists view it as a toxin. So these are secreted things, common goods, public goods. But there are exceptions to the rule, and one that I'm going to talk about today is adenosine catabolism. So the gene required for the ability of Pseudomonas to grow on adenosine codes for a cell-associated enzyme. Adenosine can be transported into cells and used as a carbon and energy source. It's controlled by quorum sensing. Di and tripeptide utilization as nitrogen sources is controlled by quorum sensing. And there are actually 2 cytochrome oxidases that are controlled by quorum sensing. Their levels go up when a quorum has been reached. So this raises the question, why regulate what I'll call a private good, like the enzyme for adenosine catabolism, by quorum sensing? It can't be to coordinate group activities. So there are lots of ideas out there about this. We focused on this gene, this nucleoside hydrolase gene. One of our colleagues in Switzerland had already reported that LasR mutants don't grow on adenosine as the sole carbon source. He figured this out basically from our transcriptomics data. We didn't even notice this in the transcriptomics data. So this became of interest to us. Now the data, I don't know, the talk's half over. So we hypothesized that a relatively few cellular processes that we call private goods, have been subsumed into the quorum-sensing regulon to allow metabolic selection against quorum-sensing mutants as social cheats. And this nucleoside hydrolase is an example of one of these. So now we can do social engineering experiments, and we sort of took a cue from Martin Schuster. He was a postdoc in my lab, and when he went to his faculty position at Oregon State, he did the first of what I call social engineering experiments. He developed a system where LasR mutants predictably emerge as social cheats in populations cooperating to grow on milk protein by using a LasR-activated extracellular protease. And this is our version of his simple experiments. So our favorite strain of Pseudomonas aeruginosa is called PA01. We use a minimal medium that has casein as the sole source of carbon and energy. So they have to use this LasR-dependent protease to grow on casein. We inoculate the medium to a certain OD. The bacteria take about 24 hours growing in log phase before they reach stationary phase, which is very convenient. You can come in every morning and transfer, keep them growing basically in log phase. So you come in every day and do this, and then we screen for cheaters at 5-day intervals. So the experiment is simple. You transfer cultures every day, and the screen is simple. We know that LasR mutants won't make extracellular protease, so we can isolate a bunch of colonies on rich medium and then spot them onto a medium with skim milk. So they have skim milk as one of several carbon and energy sources, and if they make extracellular protease, there'll be a zone of clearing around the colony. So here's a mutant that doesn't make the protease. It might not make it because it's a protease mutant, or it might not make it because it's a LasR mutant. It turns out that all the protease mutants are LasR mutants. We sort of knew this by also screening on adenosine auger. Here, the only source of carbon and energy is adenosine. So LasR mutants won't grow. So here's a wild type, makes protease, grows on adenosine. Here's a mutant, doesn't make protease, doesn't grow on adenosine. There are two functions that require LasR that are lost. This is presumptive for a LasR mutation. Sequencing showed that these all have LasR mutations. So here's the experiment, still simple. So we follow the percentage of protease negatives over time in these social engineering experiments, and they're really below detectable until 2 or 3 weeks into the experiment, and then they rise to about 40% of the population, and there's this stable equilibrium, which we didn't really understand at first, but cheaters do arise. They have a fitness advantage over the cooperators. So the population biologists explain this sort of equilibrium in mathematical terms, something about the cost of the goods versus the advantage to cheating. And this is entirely unsatisfying to me. I'm a mechanism person, right? And I don't like math. So I've taken a little bit different approach than most people in this field. I already told you about this. The protease negatives all are unable to grow on adenosine, and they're all LasR mutants. So now I want to tell you about restraint. And this is where we raise the question of why are some private goods controlled by quorum sensing? And we ask the question, what happens with casein and adenosine in the growth medium? Casein is the good carbon and energy source here, but the bacteria will use adenosine at some slower rate. So there's one publicly and one privately metabolized LasR-dependent carbon and energy source. The hypothesis, adenosine in the medium will reduce the frequency of social cheats. This will be a metabolic constraint. Quorum-sensing mutants will be penalized because they can't use adenosine. They can't cheat on the ones that can use adenosine. So here's the experiment. It's the same setup. Ajay Dandekar did this experiment. He's a pulmonary doc, so he talked about 4 doses of casein and adenosine. So in this experiment, we kept the carbon concentration the same, decreasing concentrations of casein, increasing concentrations of adenosine. If there's enough adenosine, will cheaters still emerge? Well, no, they won't. Cheaters can be suppressed by adenosine. So here's the simple experiment. Here's our control, the black line. About 40% of the individuals become cheats after a couple weeks. Anytime the adenosine concentration was over 0.5%, cheaters didn't emerge. They always stayed below 1%. So they were, presumably they were penalized because they couldn't use the adenosine. So the private good, does it have to be controlled by quorum sensing? One way to ask this question is to replace the adenosine with glucose. Glucose is an energy source for Pseudomonas. It's not the best one like it is for coli, but it's used in a quorum sensing independent way, and it doesn't suppress the emergence of LasR mutant cheats. So here's one experiment that I'll show you. And here we're using 0.25% casein. So in pure casein, cheats still emerge. So this, you might have been wondering whether the last experiment was because we were lowering casein or raising adenosine. Even at low casein, cheats emerge. And in the presence of glucose, they still emerge. Maybe a little more slowly than they did without glucose, but they still emerge. And in this experiment, they reach 20 to 30%. So the private good has to be controlled by quorum sensing. But there's no tragedy of the commons. You know, we'd like there to be, because then infections would be sort of self-limiting. There was a stable equilibrium of cooperators and cheats. Why? They should cause a tragedy of the commons. And again, I'll use my tax analogy. You know, we tolerate tax cheats. There aren't so many of them. Some of them are contributing a little bit, so they're not pure cheats. And we still get the advantage of cooperating. So they're not so expensive. But we wondered if we could increase the cost of cooperation. If cooperation becomes more expensive, then there should be more of an inducement for cheats. They should rise to a high level and cause a collapse of the population. This is what I think, well, I don't know who's proposing that we should never raise taxes now. This should be the argument. If taxes are too high, we'll cause a collapse of the population. I gave a talk like this a few weeks ago in Hong Kong, and there was a guy there from Sweden who came up to me afterwards and said, "You're wrong, because we pay incredibly high taxes and we haven't collapsed." So I don't know. But anyway, what happens if we can increase the cost of cooperation? And we wanted to do this in a simple way. We removed ammonium chloride from the medium. Ammonia is the favorite nitrogen source for Pseudomonas. Now Pseudomonas has to use casein as a sole source of nitrogen as well as carbon and energy. They have to make more protease to do this. So this is now more expensive. Will this cause a tragedy? And if it does, can we still incentivize cooperation with adenosine? So here's the simple experiment. Over here, I'm showing piocyanin production. Piocyanin is controlled by quorum-sensing. And here's what happens in our experiment with ammonium chloride and the medium. Over a period of 2 to 3 weeks, I guess this is 3 to 4 weeks, the population becomes more efficient, maybe is the word. They produce more piocyanin than the unevolved population. So there's clearly some adaptation going on during this time. If we remove the ammonium chloride, here are 3 individual experiments, piocyanin concentrations become very high. I think they were about 5 times or 6 times as high as the concentrations in evolved cultures with ammonium. This is just a pictorial of what these evolved cultures look like. And it turns out that the proteases are also about 7 times higher than proteases in cultures with ammonium. And when they get to here, what happens? So cheaters arise, they crash right through this 40% barrier, reach about 90% of the population. So 10% of the population here are making a huge amount of all these things. It's really expensive for them. And we can no longer transfer and get cultures to grow. So there's a collapse. So when we try and inoculate fresh cultures with this many cheaters, there aren't enough cooperators to get the thing going again. So what about policing? Remember, policing is cooperators doing something to hinder the success of cheaters, and this something that they do costs the cooperators. So policing has been talked about in the bacterial world. There's one bona fide example in a bacterium that's been used as a model for bacterial sociality for decades, Myxococcus xanthus, but the mechanism for policing in Myxococcus isn't known. So in our evolution experiments, there was a stable equilibrium of cooperators and cheats on casein if there was ammonium chloride in the medium and no adenosine. And again, this is a sort of a repeat of a slide I keep showing. Cheaters should cause a tragedy of the commons, but they reach an equilibrium of about 40% in the population. Why? Maybe the cheats aren't pure. This is one idea. Maybe they evolved to provide some benefit to the cooperators. So maybe there's some sort of mutualism that evolves. This would be interesting. This was a Jay's favorite idea. Maybe the proteases are partially privatized. If cells hold on to some of their protease on the cell surface, then cells that don't make protease will be at a little disadvantage. And I like this idea. Maybe cooperators police cheats. And I had an idea about how. And so I'll give you the hypothesis. I'm telling you all this obviously because this is a rare example where I was right and the J was wrong, right? So hypothesis is that RelR activates genes for toxics. I already told you that it activates the hydrogen cyanide genes. And if RelR co-activates genes for resistance to toxics, and I already told you that cytochrome oxidases go up in response to quorum-sensing, then cooperators could hurt cheats. And remember, I told you that LasR activates RelR. So if you knock out LasR, cells won't make cyanide. They won't make these cytochromes at higher levels, so they won't resist cyanide either. So we can do a simple experiment. Our same evolution experiment, starting with a RelR mutant. So this mutant doesn't make cyanide, doesn't increase levels of cytochromes in response to quorum sensing. And here's what we see. This is cheater emergent starting with a LasR mutant. So cheaters emerge, and these blue lines are independent experiments. They emerge at different times in these experiments, but they crash right through the 40% barrier, get up to 80 to 90%, and then there are insufficient cooperators to continue to transfer. Here's a control experiment, actually 3 of them all plotted on one line, with a wild-type bacterium reaching nearly 40% of the population. The cheats reach nearly 40% of the population. So this is consistent with the idea that LasR is required to activate something involved in policing. We can do an experiment with a LasI mutant, sort of a control experiment, a phenotypic complementation study. So the LasI mutant does the same thing as the LasR mutant. That's good, right? But we can phenotypically complement by adding C4HSL to these bacteria. They still have a functional LasR, so they can respond. And when we add C4HSL, cheaters arise, but they reach about 40% of the population. That's what we would want. We went on to show that LasR activation of hydrogen cyanide production specifically mediates policing. So LasR could be controlling hydrogen cyanide or something else. We took a bet on hydrogen cyanide. So you start with a mutant that can't make cyanide, and then introduce a LasR mutation into that, do competition experiments, so now the wild type doesn't make cyanide, so we would predict it can't police the cheats. And what happens? Starting at 2 different initial densities of cheats, 1% or 10%, as they rise up to be about 90% of the population, then the population crashes. So this meets the policing model. And these are pure culture experiments. Growth of the wild type is shown in the black line here. There's logarithmic growth for about 40 hours. If you knock out the ability to make cyanide, the wild type picks up steam. This is growth on casein. The wild type picks up steam after about 18 hours and reaches stationary phase after about a day. So this difference is the cost of making cyanide. So the wild type is presumably more resistant to cyanide than the cheat, but it still is expensive to make it, and it still causes it to grow slower. So on to the conclusions. Great. These are idealized homogeneous lab cultures. So that's that. Social engineering in the lab. LasR mutant social cheats emerge among cooperators. Naturally occurring LasR mutants are, as I've said here, a story for another day. I told you that they occur in cystic fibrosis infections, for example. It turns out that what these mutants are is much more complicated and interesting than it would appear at first glance. So there are all sorts of complications in how they get around having LasR. So it looks like LasR is something that's not that great for them in the context of a CF infection, but they've evolved ways to, in a lot of different ways, to express quorum-sensing controlled genes that are involved in the infection, even in the absence of a functional LasR. So again, that's a story for another day. Quorum-sensing co-regulation of a private good with a public good affords a strong constraint on cheats. If cooperation is expensive, the incentive to cheat increases and a tragedy ensues. This LasR-RelR cascade allows P. aeruginosa cooperators to police sheets. This is the first hint of a mechanism for policing in bacteria. Cooperators produce cyanide and show enhanced resistance to cyanide. So those are sort of conclusions of the data that I showed you. We're interested in this. Why do LasR mutants emerge in some types of persistent infections? That's why we're studying lots of isolates from infections. We still don't know the answer. Social cheating could be part of the answer. Maybe there's some sort of growth advantage. We know that many of the LasR mutants from cystic fibrosis patients grow better on phenylalanine than wild-type versions that they've evolved from. This could afford them some ability to evade the immune system. Our pie in the sky is that by learning about communication and control of cooperation, we can devise ways to induce a tragedy of the commons and resolve infections. We've at least learned that maybe targeting LasR with small molecules isn't the best target for at least cystic fibrosis infections. And the epilogue is really short. It's the thank you slide. This is our group. I've talked about social interactions in Pseudomonas today, but there are lots of other things going on in the lab. Mo Thomason was a graduate student here. She's now working with us as a postdoc on some very interesting quorum-sensing regulatory mechanisms. We work on quorum-sensing in Burkholderia. Members of this genus can anticipate overcrowding. They can get ready for stationary phase, and quorum-sensing mutants can't survive in stationary phase. We have a program working on plant microbe interactions in collaboration with a group in China. And Becky Schultz is a graduate student who was sidetracked on to studying siderophores in E. coli. And all of the work in my lab is funded by a grant from NIGMS. I mention this because my program officer threatened to be here today. And we get help from a grant from NIDDK that supports a core center for studying cystic fibrosis. So with that, I'll stop, and I hope for some interesting questions.
Please go to the microphone. For questions, please go to the microphone.
Okay. It sounds like what you're really working up to is what's commonly known now as biofilms. It's kind of a prelude and kind of a pretty story that kind of leads up to the new concept of biofilm, where you have an outer layer of bacteria that kind of are the active guys, and you guys got other group on the inside that kind of sit around, and then when the antibiotic comes along and tries to kill off the outer layer, the other ones go to replace them. Then this is pretty well, I don't know if it's well established, but I've seen it in the literature.
Yeah, so I'm responsible for a lot of that literature, actually, and I'm working away from that. So in the 90s, we showed that in Pseudomonas, quorum-sensing affected how biofilms developed in the lab. And sort of led a push with my colleague Bill Costerton and a colleague in Denmark to change the way that we treat cystic fibrosis patients by being very aggressive about therapy. We showed that CF infections were primarily biofilm infections. Yeah. So go ahead.
I know I get what you're going, but I can say I've actually done autopsy, at least one autopsy on a cystic fibrosis patient in the 20s a long time ago, and I remember seeing the coloration of the lungs like a bluish green, and it was just massive bronchiectasis, which, the bronchi were just like gigantic. Well,
yeah,
they looked
big,
and the parenchyma was just totally shot. Now, I would think that that type of, I don't want to interrupt their questions, but I would think that that could be, if you have your cheater bacteria and you could introduce them into it, that would have prevented perhaps that problem that I saw so graphically in the anatomic pathological correlation, no?
Yeah, so let me just say that I worked on biofilms for about 10 years, and some of the people younger people that I brought along and are on our faculty now still work on biofilms. But I got too frustrated by biofilms. You know, what we study in the lab maybe not be relevant to what's in an infection. Mike Otto, who's here somewhere, works on biofilms too and has the same frustrations. And the models are so bad that different people get different kinds of results, and it is messier than I wanted to deal with. So I've steered away from them. The idea of introducing cheaters into a CF lung has been raised by a colleague in Nottingham, Steve Diggle. And he's actually done some mouse experiments that show that cheaters can cause collapses in certain mouse infections. It would be cool. I argue that, you know, getting the FDA to approve a view putting Pseudomonas of any kind into the lungs of a CF patient is a non-starter. And there are much more interesting therapies coming along and actually now being used to prevent lung infections and cystic fibrosis.
Thank you very much.
Yeah.
Dr. Greenberg, so you said it may not be the best idea to inhibit the quorum-sensing receptor like LasR because they easily being mutated after like 2-3 weeks, as you're showing in the data. But so far, there's been numerous quorum-sensing inhibitors reported. So if this is not a good idea, do you think it may be a better idea to inhibit the transporter of the quorum-sensing? Say, in the beginning you said there's only been diffusing to the environment, but not in the transporter. But I may have seen some papers say there's a transporter on the membrane to recept the quorum-sensing molecule. But is there any the transporter to date known to transport out of the?
Yeah. So the one report I believe is that for the long chain acyl HSLs, efflux pumps help the signal get out. So the signal will get stuck in the membrane and can be helped out by efflux pumps. But even mutants that don't make the efflux pumps, still signal gets out. There are reports, one of them is in Burkhold area, that transport is absolutely required to get signal out of cells, but they're one-offs. All of the few reports are one-offs that I don't know how to put this tactfully, that I don't yet believe. So we'll see. But I don't think, my view is that you don't need a transporter to get any of these signals in and out of cells, so that transport wouldn't be a good target. What I didn't tell you about the LasR mutants and cystic fibrosis is that many of them have somehow short-circuited the LasR system by replacing it with the REL system. So the REL system becomes independent of the LAS system, which means that maybe the REL system would be a good target. Or maybe if you can target generally signal production, which we've made some effort to do, you can block both quorum sensing systems, and that might have some therapeutic value.
Thank you very much.
Yeah.
Really nice talk. Thank you, Pete.
Thanks, David.
So I've been out of this business for a while, so forgive me for naive questions. So I'm interested in LasR and RELR. First question is, do they locate ever close to the membrane, the membrane?
Okay.
First. Second, do they form dimers? Do they only form homodimers, or can they form heterodimers?
Yeah.
And the mutants that you find, are those mutations in binding ligand, or are those mutations in cooperating binding protein-protein interactions?
Okay, let's see, what was the first question?
Membrane location.
Membrane localization. I don't know. So they're clearly not membrane proteins per se. I had a graduate student in the 90s who struggled with LasR and Vibrio fischeri. It was before we even knew about LasR. And she had evidence that LasR was loosely associated with the membrane. You could shake it off, but otherwise it would go with the membrane in a separation. And so I don't know what that means. It's never been followed up with any other R proteins. We've purified bunches of them from the soluble fractions, cytoplasmic fractions of cells. They are, as far as I believe, homodimers. Each monomer binds a single HSL molecule. They're, sort of typical two-domain transcription factors with an N-terminal signal binding domain and a C-terminal DNA binding domain. This is all the work I glossed over. There's one report that came out about 20 years ago from a colleague who did some very sort of complicated fluorescence study and published a paper that suggested they could form heterodimers. But in Pseudomonas, we know that RelR is at a concentration of about 20 times higher than LasR. So, there's not enough LasR so that there are going to be any significant RelR heterodimers. And then the third question
was the mutations. Where do the mutations?
Oh, the mutations. So, two-part answer. The mutations occur primarily, they occur in both domains. So, in any independent experiment, what comes up is in a different place. Most of them come up in the DNA binding helix-turn helix. So we think those are, really, there's no hope for those. We've looked, a colleague at Washington, a CF doc, was involved in a study of pediatric patients, really following up on our idea about how to best prevent Pseudomonas colonization in children. And they looked at a couple hundred children over a period of several years and isolated thousands of Pseudomonas. And he was thinking that because LasR mutations seem to come up later after colonization, that LasR mutants might be some sort of biomarker for worsening health. So he sequenced all of the LasRs, and it turns out that they're not a biomarker, but this is like a gold mine for us, right? So we've been looking carefully at those. The mutations happen all across LasR. There are point mutations, non-synonymous point mutations. There are deletions. There are insertions, all sorts of things. About 20% of the things that he called LasR mutants aren't LasR mutants at all. They're sequence variants that still make active LasRs. So it's kind of a hodgepodge in there that we're sorting out.
Thanks.
Can we have one more person asking questions? Yeah, you.
Yeah.
I was just wondering if there were any analogs that you were aware of the quorum-sensing phenomenon in multicellular eukaryotes.
Well, do you consider yeast a multicellular eukaryote?
If you can go higher. Up
You
the
want to go
chain,
higher.
that would-
Analog. So clearly the yeast make quorum sensing signals and respond, and some of the pathogenic fungi do this. Apparently histoplasma makes something that controls virulence. Going higher, there have been reports describing quorum sensing in insects. So bees, somehow, I don't know all the details, but when bees go out and decide where to make a new hive, it involves a collection of enough pioneers in one place, and then that somehow recruits more of them. But
so that's at the level of the whole
of the
organism,
whole, you
but-
want
but
cells, yeah.
Cells within a multicellular organism.
No, as far as I know, not. And so I think I mentioned during the talk that I was in Hong Kong a few weeks ago, and I gave a talk similar to this. And that was at a stem cell meeting, and I kept asking them, do you really want me to come to this stem cell meeting because I don't work on anything close to stem cells? And they kept saying yes and yes and yes. And there's some reason that people working on mammalian cells think that there might be some sort of analogous signaling system, but it hasn't been identified yet. That is enabling growth of cells. That's the best I can do.
Thank you.
Yeah.
Okay. I want to remind everyone that there is a reception in the library next door, and also that Professor Greenberg is giving another talk tomorrow at 11 in Building 37 at the 6th floor conference room. This is part of Lambda Lunch, and everyone is invited to come to that. But let me just end by thanking Peter for an excellent talk. Thank you very much.